In E. coli, glucose is used primarily as a carbon source for energy production. E. coli takes glucose molecule inside, and this glucose molecule is used in glycolysis pathway, and energy is produced in the form of ATP. This energy is used by E. coli for metabolism and survival. For using glucose as energy source, there is no need for induction of an operon. E. coli preferentially uses glucose molecules for energy production. First, it finishes all glucose molecules in the medium. All these glucose molecules are used in glycolysis pathway for energy production. When all glucose is depleted, only then it uses other carbon source for energy production. But when lactose is present in the medium, then E. coli needs expression of set of genes known as lac operon. Products formed by expression of this operon play important role in metabolism of lactose. Presence of lactose molecules induce an operon on chromosome. Let's see the structure of this lac operon. This is the structure of lac operon. Lac operon has three different regions such as regulatory region, control locus, and structural locus. In regulatory region there is regulator gene LAC-I is situated. This regulator gene LAC-I codes for repressor protein. Control locus consists of promoter and operator site. Promoter is the sequence on double-stranded DNA, where RNA polymerase binds and initiates process of transcription. Operator site is the region between promoter and structural genes. This operator region acts as on and off switch. Structural locus has three structural genes. First structural gene is LAC-Z gene, which codes for beta-galactosidase enzyme. Second structural gene is LAC-Y gene, which codes for permase protein. Third structural gene is LAC-A gene, which codes for thiogalactoside transacetylase enzyme. Function of all these three proteins are: permase protein is membrane protein. Lactose molecule enters inside through this permase membrane protein. Once lactose enters inside the cell, it gets hydrolyzed by beta-galactosidase enzyme. Beta-galactosidase breaks down the lactose into glucose and galactose. Then glucose and galactose is used in glycolysis for ATP and energy production. Transacetylase enzyme transfers an acetyl group from acetyl coenzyme A to beta-galactosides. When lactose is absent at that time, lac operon is in off mode. Operon remains repressed by repressor protein. Let's see how repressor protein does this process of repression. LAC-I gene codes for lactose repressor protein. This repressor gene I is constitutively expressed. That is, cell continuously makes repressor protein. Transcription of LAC-I gene produces repressor mRNA. Translation of this mRNA produces repressor protein, which is a monomer. Such four monomers joins together and form a tetramer. This LAC repressor is multimeric protein. Repressor protein has two functional regions. First is DNA binding site, and second is allosteric site, which binds to allolactose. When lactose is absent at that time, repressor protein is active, and it binds to operator. Now RNA polymerase binds to promoter, and tries to transcribe LAC-Z, LAC-Y and LAC-A gene. But RNA polymerase can't transcribe ZY-A genes, because repressor is blocking the path. Result is there is no transcription of lac operon, and ZY-A genes are not expressed. In E. coli, initially there are very few lac permase protein and beta-galactosidase enzymes are present. So when there is lactose is present in large quantity outside cell, then few lactose molecules enter inside E. coli through membrane protein called lac permase. Once lactose goes inside, then this lactose molecule gets hydrolyzed by beta-galactosidase enzyme into galactose and glucose. At the same time, few molecule of lactose gets converted into its isomer allolactose. This allolactose has affinity for repressor protein. Allolactose binds to repressor protein. Binding of allolactose to repressor protein changes its shape, and repressor protein become inactive. Repressor no longer remain on operator, and RNA polymerase has full access to operator. Now path is clear for the RNA polymerase. RNA polymerase now transcribes LAC-Z, LAC-Y and LAC-A genes. Now a polycistronic LAC mRNA containing LAC-Z, Lac-Y Lac-A mRNA sequence is produced. After translation of this mRNA, three different proteins such as beta-galactosidase, Lac-permes, and transacetylase are formed. All these proteins are then utilized for taking all lactose molecule inside cell. And breakdown of all these lactose takes place in glycolysis pathway and energy in the form of ATP is produced.